Hello and welcome to my YouTube channel, AnatomyMentor. Today I'll be discussing a topic of embryology, which is the development of the nervous system. And this is actually a series of lectures, and the first part of the series of lectures is going to be discussed today, in which we are going to be talking about the basic formation of the nervous system during embryonic and fetal life. So here are our learning objectives for today. Now remember, this is a very important topic, not only in case of embryology, but also when we're talking about neuroanatomy. So today what we are going to be discussing is the formation of the brain vesicles, which are the primary and the secondary brain vesicles. And also we'll be talking about their adult derivatives, as well as the stages in the formation of the layers and horns of the spinal cord. These are our learning objectives. Along with that, we'll be talking about the formation of various types of supporting or glial cells within the nervous system, and also the formation of the spinal nerve and the role of the neural crest cells. And I'll be talking about the positional changes of the spinal cord and correlate that with the spinal tap. So let's start with today's discussion on the development of the nervous system. Now, keep in mind that the story begins with the vesicles, which are the large prominences, and these are known as the brain vesicles, which give rise to the formation of the brain. If you recall, during your lectures of general embryology, we talked about neurulation. Neurulation is the formation of the neural tube, which occurs in the third and fourth weeks of intrauterine life. Well, because of that neurulation, there is formation of a neural tube which lies in the median plane of the developing embryo. In the upper part of that neural tube, soon after neurulation, three prominences appear, which are known as the primary brain vesicles. So if we look at these brain prominences, these primary vesicles, they are known as the forebrain, the midbrain, and the hindbrain. So this is the naming of three primary brain vesicles in a craniocaudal sequence. Remember, these are the prominences of the upper part or the cranial-most region of the neural tube. This forebrain is known as prosencephalon, that's the medical word for it. The midbrain is known as the mesencephalon, and the hindbrain is known as the rhombencephalon. This diagram is representing a sagittal section through the upper part of neural tube at approximately the fourth week of intrauterine life. Now, these three primary brain vesicles are further going to give rise to 5 secondary brain vesicles. From the forebrain or the prosencephalon emerge the 2 secondary brain vesicles known as telencephalon and diencephalon. Also, you can see the midbrain or mesencephalon is not going to give rise to any further secondary brain vesicles. It will continue simply as the mesencephalon, which is connecting the diencephalon to the hindbrain or rhombencephalon. Rhombencephalon will have further subdivided into these 2 secondary brain vesicles, which are metencephalon cranially and myelencephalon caudally. So let's count the secondary brain vesicles. There are going to be 5 in number. That is telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So this is approximately 32 days of human development, and now you can see that these are 5 secondary brain vesicles. Also, you can see there is a bend in the cranial region of the developing brain, and you can see that the connection between the mesencephalon and the rhombencephalon is narrowed down at this region, which is known as the rhombencephalic isthmus. Now, these are a series of diagrams which I find very helpful, and I always ask my students to draw these diagrams in their practical copies. These are from Keith L. Moore, Langman, Medical Embryology. Keith L. Moore will show you these 3 brain vesicles, which are the primary ones, prosen, mesen, and rhombencephalon. And then it will show the further developmental stage in which the prosencephalon has formed the telencephalon and diencephalon. And here you can see telencephalon itself is consisting of two bulbous dilatations, and then the diencephalon below is continued as mesencephalon, whereas the primary brain vesicle rhombencephalon is going to form meton and myelencephalon. So this is a very simplistic diagram which is showing us the development of the brain vesicles. And I like this diagram because it also shows you the adult derivatives of the secondary brain vesicles. The telencephalon is going to form the cerebral hemispheres of the brain, which contain the lateral ventricles. The diencephalon will form a number of structures in the midline of the brain, like the thalamus, the hypothalamus, and the cavity associated with this region is the third ventricle. The midbrain is going to be derived from the mesencephalon, and within it is the narrow aqueduct of Sylvius. Metencephalon is the part which forms the pons and the cerebellum, and in association with these parts of the brain are the fourth ventricle. Whereas the myelencephalon will form the medulla oblongata, and it contains the lower part of the fourth ventricle. So this is a good diagram which is summarizing the development of the brain vesicles and also showing their adult derivatives. Now let's talk about the spinal cord. As we continue downwards caudally from the myelencephalon, we have the neural tube, and that the remainder of the neural tube is going to stay narrow. There are not going to be any vesicles over here. This is the part which is going to form the spinal cord. Now keep in mind that the wall of a recently closed neural tube consists of the cells which are neuroepithelial cells, and they form a thick pseudostratified epithelium in the wall of the neural tube. Pseudostratified, as the name shows, is the epithelium which is appearing to be multi-layered, but is in fact consisting of a single layer of cells resting on the basement membrane. This neuroepithelium, the cells in this neuroepithelium are going to be dividing and giving rise to more cells. And over here you can see this is our pseudostratified epithelium in the wall of the neural tube in initial stages of development, and the dividing neuroepithelial cells can be easily discerned by the mitotic figures present over here. And this is, you can see a picture which is taken with the help of the scanning electron micrograph of the pseudostratified epithelium, that is the neuroepithelium in the wall of the neural tube. Now these neuroepithelial cells, they're going to give rise to the primitive nerve cells known as neuroblasts. And these neuroblasts will form a mantle layer around the neuroepithelium. This mantle layer is going to later form the gray matter of the spinal cord. As you know, the gray matter is the part of the spinal cord that contains the neurons or the nerve cells. So here is then again a diagram of that pseudostratified epithelium, which is consisting of the dividing neuroepithelial cells. And as they're moving towards the lumen, you can see they're differentiating into neuroblasts, which are then going to give rise to nerve cells or neurons, which are the functional cells of the nervous system. Now, the outermost layer of the spinal cord is the marginal layer. This is formed by the nerve fibers which are emerging from the neuroblasts of the mantle layer. So marginal layer is surrounding the mantle layer. Since mantle layer is going to form the gray matter of spinal cord, this marginal layer is going to form the white matter of the spinal cord. This is the part which contains the nerve fibers of the neurons. If you look at the stages in the development of a neuroblast, initially remember it is not having any process, and it is known as an apolar neuroblast. Then two processes will appear on opposite sides or opposite ends of this cell, and over here it is called a bipolar neuroblast, which eventually differentiates into a multipolar neuron which has typically many processes on one side known as the dendrites and a single long process which is the axon. Once neuroblasts form, remember they lose their ability to divide. So adult neurons do not have the ability to divide. What are the stages in the development of the spinal cord? Well, due to the continuous division of neuroblasts, each side of the neural tube, it shows certain thickened areas which are known as the basal plates and the alar plates. The basal plates are oriented ventrally and the alar plates, their orientation is dorsal. Also keep in mind the basal plates have the ventral motor horn cells, whereas alar plates are the sensory dorsal areas. Longitudinal groove known as sulcus limitans, it marks the boundary between the basal and the alar plates. The midline portion of the neural tube does not contain neuroblasts. These are simply the pathways of nerve fibers, and these regions are known as the roof plate in the dorsal aspect and the floor plate in the ventral aspect. So here these diagrams are from Langman's medical embryology, and they're showing two successive stages in the development of the spinal cord. Now in diagram A, which is the earlier stage of development, you can see the blue regions over here are representing the thickened areas, which are the alar plates, and these are located dorsally. Ventrally, the regions in red are representing the basal plates, and basal plates contain the motor neurons, whereas the alar plates, as I told you, are the sensory regions. Also keep in mind the alar and basal plates, since they contain the neurons, they are representing the mantle layer over here, which is in the further stages of development going to form the gray matter of the spinal cord. And this mantle layer on the external aspect is surrounded by the marginal layer, which, as I told you, contains the nerve fibers of the neurons of the mantle layer. And marginal layer in the adult spinal cord, is going to become our white matter. Also, let's look over here. This is the sulcus limitans, which is the boundary between the alar and the basal plates. Also, we should locate the roof plate and the floor plate. As I told you, these are the regions which contain only nerve fibers and they don't have any neurons. So now diagram B is showing us the later stages of spinal cord formation, and you can see that dorsally there is a median septum and anteriorly there is a ventral fissure. And let's look at the central canal. The cavity of the neural tube has now shrunken to a narrow region, and now it is known as the central canal. And it is lined by these epithelial cells. And the dorsal sensory horn, which is derived from the alar plate, is now narrow in comparison to the broad ventral motor horn, which is derived from the basal plate. Now, what about the glial cells? Other than the functional neurons, there are many supporting cells within the nervous system. And in the primitive stages, these supporting cells are called glioblasts, and they are going to form the glials or supporting cells. Glioblasts themselves are also formed by neuroepithelial cells. They migrate from the neuroepithelial layer into the mantle and marginal layers. So the neuroepithelial cells, other than giving rise to neuroblasts and neurons, also give rise to the glioblasts, which will form the supporting cells of nervous system, namely the astrocytes, which are fibrillar and protoplasmic types of astrocytes, and also the oligodendrocytes. And keep in mind the remaining neuroepithelial cells, they will form the ependymal cells, which are going to line the central canal. These supporting cells have multiple functions. Astrocytes, they are involved in the scar tissue formation, and they also have many immune properties. And if we look at the oligodendrocytes, they're responsible for myelination of nerve fibers within the central nervous system. There is another glial cell in the nervous system, which is microglial cell, but keep in mind this is distinct in this aspect that it is not derived from the neuroepithelial cells. Instead, its origin is from the mesenchymal cells. Now let's talk about the formation of the spinal nerves. Neural crest cells give rise to the dorsal root ganglia of the spinal nerves. Now let's take a quick recap. What are neural crest cells? If you recall from the previous lectures of neuroembryology, during the formation of the neural tube in the 3rd to 4th weeks of development, as the neural folds they merge with each other, from the edges of the neural folds, cell population is going to emerge and it's going to scatter throughout the body and gives rise to many important structures in the body. This cell population is known as the neural crest cells. And these neural crest cells, they are so important in giving rise to many different structures in the body that they have also been known as the 4th germ layer. Now, these neural crest cells give rise to dorsal root ganglia of spinal nerves, and then they form the dorsal root neurons. Their central processes penetrate the dorsal portion of the neural tube and form the dorsal sensory root of the spinal nerve. What about the ventral root of spinal nerve? That is derived from the axons of neurons of the basal plate, which emerge through the ventral aspect of spinal cord. Remember, Schwann cells are also derived from neural crest cells, and they myelinate the peripheral nerves. So here are the diagrams which are showing us the formation of a spinal nerve. This is the dorsal root ganglion, which is derived from, as I mentioned, the neural crest cells. And here you can see that the neurons in this dorsal ganglion are the pseudo-unipolar type of neurons, which means they have a single process which emerges and then divides or bifurcates into two processes. The central process, you can see, connects with the dorsal horn, and this forms the dorsal sensory root of the spinal nerve. Whereas the peripheral process of the pseudo-unipolar neurons within the dorsal root ganglion, they are going to connect over here with the ventral motor root and form the trunk of the spinal nerve. What's the origin of the ventral motor root of the spinal nerve? Well, this is actually derived from the axons of those neurons which are present in the ventral motor horn. These axons erupt outside, and they outside the spinal cord, they emerge and they join with the dorsal sensory root to form eventually the trunk of spinal nerve. So this is the formation of the spinal nerve. And eventually, remember, there are 4 functional regions of each spinal nerve, and you can see over here they are represented in these different color coded areas. And you can see over here that we have somatic sensory and visceral sensory, and then we have visceral motor and we have somatic motor. So these are the 4 functional representatives of each spinal nerve present within the gray matter of the spinal cord. Now we're going to discuss the positional changes of the spinal cord that take place during development. This is an important point to understand, that there are going to be positional changes of the spinal cord in relation to the vertebral column during intrauterine life. So here we can see the 3 stages of development, in which A is showing us the 3rd month of intrauterine life, B is representing the 5th month of intrauterine life, and C is the newborn. So let's look at the earliest stage. Initially, the spinal cord and the vertebral column, they end at the same level. The spinal cord is present throughout the length of the vertebral column. And in the later stages, you can see that the spinal cord is now shifting at a higher level. Over here, it was extending down to the coccyx, but over here, it has now shifted upwards to the sacral level. And over here, you can see eventually in the newborn, the lower end of the spinal cord, which is known as conus medullaris, ends at the lower border of the 3rd lumbar vertebra. So there is a shifting upwards of the spinal cord. And here you can see are the nerve roots which are emerging from the spinal cord. And as they are exiting from the intervertebral foramina, they form a bunch of, you can see, fibers over here, which is known as cauda equina, which literally means horse's tail. Also, you can see that the phylum terminale is the pia mater, which is also extending downwards, and you can see its distal attachment. So initially, although the spinal cord extends the entire length of vertebral column, but it is due to the faster growth of the vertebral column and dura mater that the lower end of the spinal cord shifts to a higher level. So this shifting of spinal cord to a higher level is a relative shifting. In neonates or newborns, the spinal cord is going to end at the lower border of 2nd or 3rd lumbar vertebra. And in adults, the spinal cord is going to be further shifted upwards, and its termination might be at the inferior border of 1st lumbar vertebra, or it may even be as high as T12, but it might also be as low as L3. So there is a slight variation in this aspect. Because of the disproportionate growth, when we're saying disproportionate, we mean difference in growth rates of the spinal cord and vertebral column. The spinal roots, which are from the lumbosacral segments of spinal cord, they extend obliquely to exit their respective intervertebral foramina, thus forming, as I showed you, the cauda equina. The dural sac and subarachnoid space, they usually extend to the 2nd sacral vertebra in adults. whereas the pia mater, which attaches to the coccyx, that is the C1, first coccygeal vertebra, forms a thread-like extension which is known as phylum terminale. Now let's talk about the clinical application of this knowledge. You just found out that the lower end of the spinal cord is at either the lower border of L1 in the adults or lower border of L3 in the infants. So if you understand the importance of this level, then you can apply it in a clinical scenario in which lumbar puncture is a procedure that needs to be performed. What is lumbar puncture? It is the insertion of a needle into the subarachnoid space to collect the cerebrospinal fluid, and this collection of the fluid can be therapeutic or it can be for diagnostic purposes, and the position of the patient can be in the lying position or it can be in the sitting position. Now the important thing is that the needle which is inserted into subarachnoid space, it does not damage our spinal cord. So since we know that in adults, the spinal cord is going to end at the lower border of the first lumbar vertebra, we can safely insert this needle for lumbar puncture between the fourth and fifth lumbar vertebrae. So this knowledge helps us in inserting the needle and knowing the site of insertion. And of course, in children, in infants, in that case, you know that the point of insertion is going to be further lower down since their spinal cord ends at the lower border of L3 vertebra. So over here, typically the space between L5 and S1 might be preferred for the lumbar puncture. So this concludes with our first section, our first series of discussion on the development of nervous system. And if you found this lecture helpful, then kindly like and subscribe to my YouTube channel, which is AnatomyMentor. And I would also like you to listen to the remainder of this lecture, in which I'm going to discuss in detail the formation of the brainstem and also the formation of the cerebrum. So you can simply press on my picture, click on my picture, which is going to appear at the end of this video presentation, and you will be guided through the process of liking and subscribing to my YouTube channel. Have fun studying. Take care. Goodbye.